Welcome everyone to a new sort of session of combustion webinar. Actually, a couple of weeks ago, we restarted for the fall semester. This is my name is Amir Farouk. I'm from Kaus, Saudi Arabia, and it is my immense pleasure to introduce today's distinguished speaker, Professor Phillip Westmoreland. Phil requires no introduction. He's very well known in the combustion and chemical kinetics community. He has made us a number of very important and crucial contributions to the field of chemical kinetics and combustion chemistry. He's currently a professor at NC State, and we are very grateful, Phil, for you taking out the time to give us this lecture. And the Zoom floor is yours to start your presentation.
Great, thanks very much, Amir. What I'm going to talk today about, the advertised title is that we're going to be talking about combustion fundamentals for burning and making fuels. What I'm going to emphasize is the chemistry. I was giving a seminar to one department, and the head was saying, "Oh, don't call it chemistry." People run screaming from the room. And yet, we're all pretty used to dealing with molecules. In fact, one of the themes that I want to look at is how it's a lot simpler structurally than it may seem just looking at the mass of lots of reactions that are there. But to begin with, plainly, what we want to do is to consider what are the differences relative to conventional hydrocarbons. We've got a lot of technology, we've got a lot of science built up around that. Now we're taking plants, converting them into different sorts of biofuels. So the first general sort of qualitative question is, how do they burn? You know, what's the nature of it? What are the advantages? What are the disadvantages? Then I'll spend a large part of my time talking about how we can do that more quantitatively, measuring chemically how they're burning, and then modeling. And that modeling part, I'll transition a little bit to talk about some general ideas about how combustion chemistry can be simplified a lot in terms of our description and our understanding of it, and how that maps over perfectly well to the oxygenated fuels that are characteristic of bio-oils. So finally, I'm going to step in and talk a little bit about how these biofuels are actually made, because a lot of the same people who are studying combustion are studying how the fuels are made. And it's fortunate. I mean, one of the reasons for doing it is many of the same types of reactions occur. So first, what are they made of? Generally speaking, we're talking about sap of some sort, or seeds and the oils that come from them, or woody solids. They're very diverse chemically. So if we're talking about a sap, there's lots of things in it, mostly water, simple sugars. Maple syrup is a product in some of our countries that is widely used, basically a concentration of sap. When you talk about seed oils, you're getting to things that seem closer to the sort of materials we're used to using, oily materials, often with long hydrocarbon chains as well. I'm going to focus, as most of the work is focused on woody or lignocellulosic solids, so-called because they are made of lignin, of cellulose, hemicellulose, and water. Let me show a short cartoon here, an image, while these lignocellulosic materials, which might be wood itself or might be straws or the like, they're essentially made of plant cells. Once you dry it, it's mostly the plant cell walls that are left. And the plant cell walls, in a cartoonish fashion, are made of fibrils of cellulose, these microfibrils that are surrounded by an amorphous hemicellulose, made significantly of the same sort of thing. They're both polysaccharides, polymers of biological sugars, and then wrapping around them in some sense, sort of the way that a wiring insulation will wrap around a wire. Lignin surrounds it and provides a barrier protecting the hemicellulose and cellulose in nature, but of course we want to get at it. We maybe want to get cellulose to make paper or the like. So the lignin, hemicellulose, and cellulose are dominant pieces there once you've dried it out, but not the only thing. In any event, as I said, these microfibrils are crystalline materials, the biopolymers that are stacked up, and they're polyglucose. So if we think about how to burn them, well, we can dry them, burn them directly, or we can convert them. We convert them, say, by torrefaction to a solid char with a high energy density. We can convert them to a gas, perhaps by gasifying them to carbon monoxide, hydrogen, and a lot of other things. Or we convert them to liquids with high energy density. That's where I'm going to focus. For a number of reasons, that's a form of combustion we know that is going to be really, really persistent for a long time because of the portability of liquids and the very high energy density. At this point, still very high relative to batteries. So looking at liquid biofuels, their pros and their cons, and the advantages that make us consider them, the overriding advantage is that they are renewable. We can grow new plants, and it's potentially sustainable. We get more energy out than we put in. We can keep it going. Furthermore, biomass uses CO2. It consumes carbon dioxide and generates oxygen. Of course, that's good. From a combustion standpoint, the fact that these oxygenates are less sooting, of course, is a great advantage. On the other side, we use water, maybe fertilizer and fuel for agricultural equipment to grow that biomass. And for a number of things like ethanol from corn, it looks like in most cases, it is costing you more in terms of energy than you're getting. Plus, water is a precious resource. So is food. And making fuels from food crops in a hungry world is difficult to defend. We get carbon dioxide still when we burn it, of course, as well as other pollutants, some of them fairly new pollutants, more aldehydes in particular. And of course, it's lower energy density than hydrocarbons because in a sense, it's partially pre-oxidized. And along with that, they must be refined, maybe to remove the oxygen to have a drop-in fuel, but that's very costly. But at least to get a specified range of molecules that we would like to be able to use, it becomes important to do that. So the eyes have it, the advantage there. If we're going to burn fuels, biofuels have a lot of potential. And it's pretty clear we're going to burn fuels. Chemically, the mechanistic differences between biofuels and hydrocarbons are important from a practical sense. So what we found, though, is that using experiments, using theory, we see that the individual combustion reactions have very strong similarities. The outcome can be quite different. Having oxygens in the fuel structure means it's not just a simple, relatively simple pathway that we can go down, but there's more complexity because, so in this case that I show you here, tetrahydropyran is the base ring of glucose. So it's a good one to compare with. And the model that I developed on that my group and I had developed, says that the first thing that can happen is you pull off a hydrogen, but it's unlike cyclohexane, where the hydrogens are all alike. You've got 3 distinct positions to draw from. We'll come back to that later. But for now, let's just say we're going to consider both aspects of that. And we're going to first consider experiments. How do we get data? Because I do a lot of modeling. I use a lot of quantum chemistry and reaction theory, but the data are what I would like to be able to refer to. So classically, lavender premixed flames have a lot of advantages. We have a lot of experimental tools at our hands. But with lavender premixed flames, one, it is really combustion, which has the good part that's really combustion, the bad part that is all the reactions that are going on in combustion. But there's kinetics of them. There's thermochemistry, there's the transport, and of course, it's at high temperature normally. So the laminar flame speed is going to be the classic way that these flames have been studied at the base level, because through different ingenious experimental means, it's been possible to measure them. But if we think about what we're measuring, it's really that you're seeing a flame burn into an unburnt fuel-air mixture, leaving burnt gases behind. There's a flame front, and there's a characteristic speed that goes at. The laminar flame speed, here's Ed Law's definition of it, the steady propagation of a one-dimensional planar, maybe, or maybe not locally planar anyway, and ideally adiabatic wave relative to a stationary combustible mixture in the doubly infinite domain, infinite in both dimensions. If we look at the middle part of this figure, though, you see we go from the initial temperature T0 and we rise to some burn temperature in the limit where it's adiabatic. It turns out that quantity is a thermochemical property. We can predict that without access to any rates at all. But the products, it's definitely a rate process. The unburned gas has reactants and maybe diluent in it. We form intermediates that may or may not persist, but certainly the products persist there. And we see these different types of reactions, chain branching reactions, hydrogen diffusion and heat conduction moving into the unburned gas, so that characteristic flame speed is a powerful tool. And for the last 40 years has been one of the key tests that we use for detailed reaction mechanisms. It's most sensitive to just a few reactions, particularly the key sort of reactions like H plus O2, the chain branching reaction that gives us O and OH, CO plus OH, and of course, fuel attack. So to get detailed speciation, to get the species, the chemical species that are present as reactants, intermediates, and products, a really powerful tool is to use molecular beam mass spectrometry, or MBMS. We can use it on different types of reactors. There's been a lot of terrific work done by many people around the world since Hohmann and Wagner in the 70s really developed this, even back into the 60s. There's a standard burner even. You can buy a McKenna burner to do this, but the gist of it is this. If we look at this cylindrical burner here on its side, then the freely propagating flame that I just showed you on the other side, okay, flipped, it's burning into the unburned premixed feed of fuel, oxidant, and diluent. And the burner acts as a flame arrestor. Okay, there's some heat loss, but it allows it to be steady state, one, and very flat. So long as you're close enough to the burner, heat loss is fairly uniform. And if we sample along the middle, we can get a profile, an axial profile of composition. Here is a McKenna burner with a flat flame burning on it. But of course, what we want to do is sample that. So sticking the burner in and the flame against a sampling probe with specific designs worked by a number of people, notably Joan Biordi, who was at the Bureau of Mines in Pittsburgh. We can sample the gas, we can expand it, we can cool it dramatically, and expand it to free molecular flow where molecules aren't colliding and thus are not reacting bimolecularly. Cooling it, well, it depends, but anywhere from 10 to maybe 150 Kelvin. So cold enough that reactions, unimolecular decompositions are going to be rare as well. with using a second nozzle, we skim out the core of that expansion and pass it through as a beam of molecules streaming towards the entrance of a mass spectrometer. Typically, to get the flame thick enough to see, say about 10 millimeters thick at 20, 15, 30 torr, we have to go down to 15 to 75 torr to do that. The expansion then goes down 4 orders of magnitude or so, order of 10 minus 3, and then down 10 to the minus 7, 10 to the minus 8 for the mass spectrometer. We've developed a lot of data in my particular group and in a big collaboration that worked at Lawrence Berkeley Lab, gathering information on combustion of oxygenated molecules, notably alcohols, ethers, and esters. Vikram Seshadri, shown in the picture here, next to the apparatus that we rebuilt after I moved to NC State in 2010. There was an accident with the equipment that I was moving from UMass Amherst to NC State, but there, the type of mass spec that we're using, and it's the sticking out at an angle from the main angle of the system. In this picture, it's a quadrupole mass spec. And we're doing electron ionization, boiling off hot electrons to ionize molecules, then using electrical fields to separate the groups of ions into individual masses. We're very carefully ionizing down near the threshold so we don't fragment and break up the molecules, something that in much mass spectrometry you're trying to do. Here we're trying not to do that. So I mentioned the flame team that we built starting in 2001. Terry Cool, Andy McElroy, and I were called together by Tom Baer, who was in charge of the chemical dynamics beamline at the Lawrence Berkeley Lab. I talked with Tom back in the 90s about the attributes of photoionization. Andy had done a lot of work with that. Terry had done a lot of work with that. And Andy had built a molecular beam mass spectrometer, and I'd worked with him on that and was out there working with him when Tom called this meeting. What we created was a combination of time of flight mass spec with using the advanced light source, a synchrotron, to do vacuum ultraviolet photoionization. 2 advantages. One, the energies that we're talking about are in the energy range that we need to just ionize, not fragment, to just ionize. Also, because it's a very bright source, we can get enough photons and we can tune the energy that we're using to do that ionization. So we can scan through and detect the onset of ionization for different species and add that information to the masses that we have. We can also do very high resolution mass spec. A lot of that was advanced by Fei Chi, who participated in building this apparatus here. And then Katarina Cozy-Hunghaus, Craig Taches, Nils Hansen, Tina Casper, Min Wong, and many others have been part of this flame team that worked on this for a period of 15 years or so. It's now mothballed. Sad to say, it's expensive. But again, you look at this inset picture, you see the sampling probe, you can see the skimmer tip inside here as we're moving in. The McKenna burner is actually shown in the sketch, the beam going through and intercepting the start of the time of flight mass spec. So it's a really powerful basis for testing biofuels. And I'll show a few tests here. Start first with simple dimethyl ether. I show both a stick diagram over here and a more molecular diagram. The molecular diagrams can be powerful, especially if you can rotate them in 3D and get a sense of the 3-dimensionality of the molecule, but they get awfully crowded fast. Nevertheless, we're doing dimethyl ether with a near stoichiometric and a rich flame. I'll show you here the data for major species from a 1.68 equivalence ratio dimethyl ether flame, diluted about 50% by argon. The top reference is the one this came from, the 2006 Combustion Symposium. But you see a lot of characteristics of flat flame studies in general. You see the oxygen going down, dimethyl ether going down. You see intermediates like hydrogen coming up. You see water, carbon monoxide, CO2, and water. The curves here were with a mechanism that we devised and have improved over the years. Nevertheless, you see that there is remarkably good agreement in shape. Magnitude is pretty good even for this early mechanism. And there are uncertainties in the calibration factor that we also have to take into account. But when we see shape changes, that can mean, so for example, look at carbon monoxide showing a more steep decline. This is not, some of us use plug flow reactors. This is not a plug flow reactor. There's significant diffusion, in particular, backward diffusion towards the burner. So initially, even though oxygen and dimethyl ether are dropping in terms of mole fraction, their mass flux is constant. They're not being reacted yet. What you see is a dilution by back diffusion of material in there. And you can see that just looking at the hydrogen, the CO2, the carbon. It's actually non-zero at the surface, which is physically realistic. The so-called Danckwerts boundary conditions were taken into account to recognize that when you've got a region where things can diffuse, like the open flame, compared to a region like the inside of a burner where they can't, then you're going to have the convective flux in equals convective flux minus the diffusive flux coming back to it. We can look at many intermediates here. I show a sampling of them, including some radicals that are here. Here's OH, for example, methyl radical HCO. And again, agreement, and again, this is the early 2007 mechanism, is already quite good. Well, why is that? It's because we could draw from such strong analogies. It becomes much more complicated when we go to Tetrahydrofuran. Again, I showed it to you before. It's like a cyclohexane ring, but with an oxygen, an ether oxygen in the middle. You can see the molecular diagram starts to look really, really crowded, but we can switch back and forth, whichever serves our purposes best. The molecule is at low temperatures, it's wrinkled into a chair position, like I show, or a boat position where the ends flop up towards the top. Dynamically, it's nearly flat. And we see here that the top line is temperature, so that is should not be fitting any data. It's the experimental temperature measurement with laser-induced fluorescence. And you see quite good agreement by this point in 2013 that we're doing with the model is corresponding very nicely to the data, which says a lot about how we were improving in our ability to construct these mechanisms. Again, a lot of species, including different radicals that are present. There's our methyl again, our formal radical, and let's see. Yeah, so again, that's from the 2012 Combustion Symposium, so you can look that up. What I'd like to do is to shift real quickly and to talk, though, about the structures of the mechanisms, because we can really leverage that. If we take just a simple methane mechanism and use the structure of it, where we go from methane to carbon dioxide, the Varnats and others developed, Varnats did this string showing mechanism, say, well, it goes from methane to methyl to formaldehyde to formal to carbon monoxide, carbon dioxide, even if all we see is fuel and air goes to carbon dioxide and water. They had put together individual steps that occur, and to the uninitiated, it looks like just, okay, this is a string of numbers that I've put in. We have simplified rate expressions. We say the rate is going to equal to an Arrhenius expression, rate constant, times concentration of the two reactants, or maybe a more complicated von Kuai sort of form with a T to the B type term in here to capture a wider range of temperature. But the principles behind these things, the fact that they are not only plausible, but accessible sort of ways of describing these are really best illustrated by teasing the individual reactions apart. And we'll see that these same steps can be applied to the oxygenates. For that matter, from formaldehyde on, we are talking about oxygenate chemistry. So if we look at them, the powerful insight is that we can break down all combustion reactions into three types. Abstraction reactions, mainly hydrogen, sometimes halogens being abstracted. We can look at association reactions of combination or addition and their reverse. Reverse step is decomposition. Or we have isomerizations, whether we're talking about radical chemistry, ionic chemistry, or molecular chemistry. Molecular chemistry is mainly paracyclic, and I'll come back to that in a minute, because paracyclic chemistry happens sort of in the middle of some of these reactions, but well, I'll come back to that. So the first step, the methane goes to methyl, is an abstraction. Here's an OH group pulling a hydrogen off, and we get that. Well, there's a transition state where the hydrogen is just shifting from remote from the CH3 group, but not yet part of the OH, or maybe you could argue it's part of both or part of neither. We can take that hydrogen abstraction, we can look at energy diagrams, we can characterize the barrier in energy that it takes to actually grab that and pull it off, and can see that here in this case about 19 kilojoules of barrier, but it gets you about 64 kilojoules when you go to products. Well, you look at the rate constant data from the NIST database, and you can see it's not a straight Arrhenius curve, although it may be fitted locally by one. It's got this curvature, and that's why the T to the B type term may be added in there to be useful. So the abstraction, very common, and some reactions may look like abstractions, but they're not. So we have to deal with that sort of question too. The classic one is really methyl plus O. There was a lot of confusion in the 60s and 70s about how is methyl oxidized. Oh, methyl's a radical, there's not much there. It has to react with something that's high concentration like O2. So methyl plus O2 goes to formaldehyde, and OH was the assumed choice. And then Von Duren and Von Tigelin showed with molecular being mass spec that no, it's methyl plus O, and it makes H plus formaldehyde. That was an empirical observation, but it was very striking and very persuasive. Through a different lens, we could look at that and say, well, there's no way you can look at that as an abstraction. Is oxygen hitting it and driving off an H? Well, not exactly. What's happening is you're actually doing a combination to make a methoxy, a CH3O radical, something that you never see as a part of this reaction. Why? Because the energy, when you do this combination type reaction, and the beta scission that happens from the methoxy is accelerated because of the energy difference. You come in at a high energy, and the fresh methoxy that you make is not something that you would have at thermal temperatures. It's very energized. It's energized both by the thermal energy of the reactants, but also by the chemical energy relative to the thermal CH3O. Jargon that was invented in the 60s was to say it's chemically activated, that is thermally activated, but with chemical energy as a booster. And you can just see by the relative heights of these energies here, once you make that, it's way downhill to just boom off and make H plus formaldehyde. Indeed, looking at the kinetics, there's some scatter in the data, but look, this is 10 to the 13.8, 10 to the 14. There's, it's pretty tight. It's showing as nearly independent. Thomas Just work showed at very high temperatures, it starts to drop off. And we can understand that through understanding chemical activation, because this excited species can also have a chance of popping back apart to methyl plus O. And at very high temperatures, where you've got higher energy states of this thing, some of it can pop back apart. Most of it still wants to kick on. If we still had an ideal gas at 10,000 atmospheres, that's the point where we would be seeing methoxy. So the point is that it's so stacked against methoxy ever showing up that we see H and formaldehyde essentially instantly after we have methyl and O reacting. So this idea of chemically activated dissociation decomposition, or so-called reaction over an energy well, potential energy well, is really powerful throughout combustion, because throughout combustion, there's an interplay of temperature and chemical activation. H plus O2 is a great example because we write it in mechanisms as two different reactions. H plus O2 goes to O plus OH. H plus O2 goes to plus M, goes to HO2 plus M. But in fact, in all cases, it's H and O2 combining to make HO2, but again, it's not thermal HO2. It's energetically excited into a number of different energy states depending on how hot it is. And that has to be collisionally stabilized down to HO2. So that HO2 can fall back apart to H plus O2. And at low temperatures, it'll get stabilized as HO2. You raise temperature, and it starts to say, no, I'd rather fall back apart. And then at higher temperatures, that hot HO2 explodes into O plus OH. We see that in the data. Looking at the H plus O plus O2, it's writing it as a third body reaction. We see that H plus O2 traps the H and thus keeps it from propagating combustion below 1,000 kelvins. But above 1,000 kelvins, we propagate, plotting it on the same scale, just plotting the H plus O2 goes to HO2 at a specific density, P over RT, for 1 atmosphere of argon. You can see there's a crossover point at about 1,000 kelvins from predicted numbers and the data here. You can see at high temperatures from Varnak's classic analysis, looking at the data that he had at that time in 84. But you can see the scatter of data here. That's all the chain branching below 1,000 kelvins. You're getting HO2. In effect, chain terminating because HO2 is so much less reactive than the H atom is. So this chemical activation idea, the fact that here and with methyl plus O, we saw that you don't even see the intermediate because it is so short-lived. So we go to the next step, it's hydrogen abstraction again, OH pulling off a hydrogen from formaldehyde. We go to the next step and it's either decomposition, the H can be kicked off from the HCO, formal radical, or it can be abstracted by O2. And then we're back to chemical activation again for the reaction that releases so much of the heat in combustion, O plus OH plus CO. So here just showing the basic reaction, but then showing an adduct. Again, it's energized, it's hot, and but the exit channels are about the same going in, going out. So there's some competition there. But if you go out to CO2, it's very exothermic. And here's the kinetics. You see, not very Arrhenius at all. This curvature, yes, what we see is that the rate constant actually is dropping a little bit and then takes off like crazy. This is a consequence of the chemical activation that's going on. So the essential insights that we get, this is a quantum chemically derived chemical energy potential, shows the more complicated details, for example, the OH and CO going to H plus CO2, the plateaus at the start and at the end here. We actually go through a van der Waals complex at the start, but we scoot right over that. We actually make a trans structure initially that can interconvert to a cis structure, but this is so low below that they are in near equilibrium there. And for that matter, we can make an OCHO type structure that's there that also decomposes. So we can tease across those different points and use those principles for oxygenates. If you look at the dimethyl ether we looked at it a minute ago, what's the first step? It's hydrogen abstraction. You have, in this case, a methyl that's being produced initially just by decomposition, breaking off a CH3 group. Methyl pulls it off, and you get a radical here, but the radical can decompose very rapidly, stretching off that CH3 group, giving more CH3 that then turns around, recycles, makes more radical, decomposes more, and then you're into formaldehyde chemistry and methyl chemistry that we talked about before. So this notion of building on top of the information that we have, a hierarchical mechanism, but using these principles, abstraction, association, decomposition, or isomerization being the only reactions that we have if we take chemical activation into account. Even though tetrahydrofuran is a lot bigger, it's the same principles. We can do an abstraction, then we get beta scission of the radicals that are formed, either to kick out a hydrogen to make ring species or opening up the ring, and more abstractions, more beta scissions, and then you get to small molecules that you can follow with. Hai Wang and others have promoted the idea that in a lot of chemistry with the big molecules, what you're going to do is go through these sort of really pyrolysis type chemistries in some cases, breaking down until you get to small molecules where you can go to detailed chemistry, where things like pressure dependence might be more important. For more detailed summary, you can look at this 2010 review article that has so much of the data that we got from the Lawrence Berkeley Lab experimental system on oxygenate combustion, and talks about the mechanisms that are there too. So I commend that to your attention, but I want to shift gears. Now I want to talk about how do we get those biofuels, and take just a few slides to step through talking about how fast pyrolysis of woody biomass is of particular interest. Okay, we can do things like, let's see, let me step back again. We can do things like fermentation, other sort of destructions there. But as it turns out, right now, one of the most practical ways is just do a fast pyrolysis and get the molecules out that are formed, try to quench them to keep them from making gunk or solid. And again, starting with cellulose, that's a really good choice because any piece of paper we might have is pretty pure cellulose. It's a very important part in terms of mass as well. And it's very chemically very regular. It's a polyglucose. And you can see from this top view that we have this tetrahydrofuran 6-membered ring with OHs and a CH2OH alternating sides, and a bond in between the two, the glycosidic bond. But what you're struck by looking at this is the remarkable amount of symmetry that's there. That symmetry is created by the hydrogen bonding, but if we take this structure and now rotate it along the axis, you can see as it rotates, you can see this symmetry is present in the plane of the glucoses as well. When you get to this final structure, you see the H's sticking up. And these are OH groups. Hydrogen bonds are formed by OH groups coordinating with other OH groups, with other polar groups. And that makes this polymer organized in a crystalline fashion very strong. That's why we can tap on wood. And it's pretty strong stuff. And again, it's because of these cellulose microfibrils. Chemically, though, what it means is that we have to heat it up enough that the fibrils begin to melt. And those hydrogen bonds, those many hydrogen bonds, have to allow it to become a melt. So the tests there, the measurements that we do are really crucial. We need to find out what are the rates that volatiles are given off, and we can use thermogravimetric analysis, where our unit also has differential scanning calorimetry to look at reactions that don't involve volatile loss, but that do involve heat of reaction. Main tool, though, is a flash pyrolyzer. This is a pyroprobe pyrolyzer that we're using here, with the key analytical instrument being two-dimensional GC mass spec with time of flight mass spec data. One chromatogram, I think, makes the point pretty quickly. The amount of detail that you get out is pretty extraordinary. What you're looking at in the two-dimensional plot, we actually go through one column, and then every periodically, we will, about a 7-second cycle, we will freeze the output of that result and then suddenly release it into a secondary column. So the primary axis is the primary column, the second axis is secondary column, which can be a different type. And the colors here in the two-dimensional plot are like a contour plot. You can see in the inset, just magnifying this region here, the many different species that are showing up. The second column can separate things that weren't separated by the first column. Because you've refocused it, you've got a different separation. So in this sample from a whole biomass pyrolysate, we got 700 different identifiable species compared to conventional GC mass spec where we had 250. Furthermore, the identities of the species, one species that stands out dramatically is levoglucosan. I'm trying to draw a sketch of it over here, but you see this 6-membered ring, this Tetrahydrofuran type ring here with OHs, but with a bridging CH2O. We very quickly, other people had seen that's very characteristic of cellulose. So how can we tease out how cellulose breaks down? First thing is not to use the whole biomass, although at the end to put it back together. Let's just do it on cellulose. And in this 2009 paper that George Huber and I did, we were able to do the flash pyrolysis and do separations with liquid chromatography mass spec and find out that this levoglucosan appears first at the end of a strand of cellulose, that is a cello-in-san. Let me show that. It's easier to show what it means. Here's our cellulose, sort of just a stick figure version with these glucose rings, a polyglucose. And then what we were seeing was that we were forming a glucose attached to a levoglucosan, a diglucose attached to a cellobiose, attached to a levoglucosan, a cellobiosan, triosan, up to 7 in this group here. So it's pretty natural for us in this paper to speculate that maybe this was a clipping of the longer chain, and that maybe what we were doing was breaking it, and instead of having radical chemistry, that we're doing some sort of molecular chemistry that would convert the bridging oxygen into a CH2O that's not bridging between parts of cellulose, but bridging within, that is levoglucosan, that can make other species and break down. So that was our hypothesis, that the scission step is not radical, but it's paracyclic. So quickly comment on what paracyclic means. It's a molecular transition state, molecular association decomposition, because the transition state is cyclic and it happens with electrons around the perimeter of a ring. It's pericyclic. Other names for it as well. They're also frequently called concerted reactions because these changes that we're talking about are in the transition state. They're very transitory. So it's as if they're going all at once, that is in concert. a Diolsalder reaction is something the combustion community is familiar with. Irv Glassman in the 80s was really focused on, could we understand the formation of benzene by the reverse Diolsalder reaction, having ethylene react with butadiene or acetylene react with butadiene to make a ring species. You see this sort of ring electronic structure being formed in this electronic diagram that's here. But the animation, I think, really tells it pretty well. And the fact that it's very specific, you have to attack from the side to come in there and have the pi bond that's in the ethylene react with the pi bonds in the butadiene by coming down on top of them. So we ended up using computational quantum chemistry, relying on basic density functional theory to find transition states, and then CBS QB3 and higher level calculations to see how it was formed. So here I show the glucose with OHs stripped out just for clarity, looking at the different types of carbon. The carbon 1 is the one that bridges across the glycosidic bond that bridges from glucose to glucose. Carbon 6 is the CH2OH. And what we found was that we can pop that off. We can see an internal reaction, a four-center transition state of this sort right here that eliminates the water and makes that bridging compound. But the winner, the real fast one, is a bimolecular transition state where at water or any OH group that's in the system, and these have lots of OH groups on them, can catalyze the reaction homogeneously by allowing a hydrogen to be transferred from the OH here in carbon 6, while the water donates its original hydrogen to the O that's in the glycosidic bond, breaking that bond and forming a bond between the oxygen and carbon 1. That's a little hard to see, especially if you're not used to seeing this. So let me put it in 3 dimensions. The animation will make it much easier to see. So again, here's our OH stripped molecule, but I'm showing this bridging group. Again, this H atom could be another whole glucose. We'll just show it as an H. Carbon 1, carbon 6 with its OH, and let's go. Put in a water, and you see the water will donate, but let's look at it happen. Well, boom, this OH is leaving and it's going off as another water. This HOH, again, could be a glucose OH or something else, but it leaves. So we start with the water, we end with the water. Well, with an OH group, and we generate the OH group, and we end up with this bridged compound. So the idea there's this self-catalysis is very powerful. Suddenly, worries about how can this happen by radicals are alleviated. And it turns out, again, it is the OH, not the R in the catalytic ROH. The activation energies don't really change if you use water or methanol or ethanol or isopropanol as the hydrogen donor there, as the OH group. And there are lots of OHs present in glucose and cellulose. So this has recently been confirmed by experiments from University of Minnesota. But we went on and began looking at hemicellulose. Hemicellulose is also polysaccharide. You see these characteristic pyran rings here, but it's got side groups as well, and it's got other things beside glucose. In particular, the base here in blue is a general glucan, but we've got a xylan. It's made up of xylose, another simple sugar that doesn't have a CH2OH group on it. So by looking at that, and again, taking things like monomers, but as well as dimers and polymers, we can take these different sugars that are almost the same. Just glancing at it may be hard even to see. The difference between D-xylose and D-arabinose is the position of OH relative to the rest of the ring. The difference relative to fucose is that you've got a CH2 group over here on the side, but it's a methyl H on the same carbon that's happening right there. So we get lots and lots of different species. We can tease out in particular that acetaldehyde is a major species. We can tease out other species as well and put together a mechanism by which this is all occurring. The simple structural perturbation may be a little bit easier to see here, but you've got these two hydrogen atoms on position C5 versus H and methyl, and then you've got the stereochemical position switch in the arabinose compared to the xylose. Experimentally, what we find, glycolaldehyde is a lot higher on xylose and arabinose. Acetaldehyde dominates with fucose. And we then related that to more of these self-catalytic decomposition reactions, paracyclic reactions. And yes, you can get to radical chemistry as well. So that's a good point to come to and say that, okay, in conclusion, the idea of how do biofuel oxygenates burn is much better understood now with work developed over the last 30 years. But a key aspect of that is understanding how the elementary reactions occur and doing generalization and analogy. That sort of analogy is the basis of automatic reaction mechanism generators like RMG or Genesis or X-gas and Polymer's model that automate the process. Second point is really that the formation of the bio-oils from cellulose and hemicellulose is through these self-catalyzed reactions, as we discovered in 2012. Other groups were beginning to find, three other groups in particular were beginning to find there were molecular reactions, but we have been able to show that it's the OH groups that were handling it. And by understanding that, we could add blending of fuels, combustor operation, and so on. Those are the big ideas that I'd like to share. I, of course, do want to very gratefully acknowledge the students and other collaborators and the financial support that DOE has given me through the years, NSF, and using a lot of supercomputer resources to do these calculations as well. I enjoy spending this time with you and hope I see in the chat box there's already one question to talk about, and I'm open to more of them as well.
All right, thank you very much, Phil, for your talk. Much appreciated. It's also was very good to see that you touched both on the utilization as well as production of these biofuels, and how the same sort of techniques can be applied to understand both aspects there. Maybe taking advantage of me being the chair, I'll ask the first question. And this is more of a philosophical question. So a few years ago, I attended a seminar by Professor Hartmut Michael, who is from Max Planck Institute. And the title of his talk was The Nonsense of Biofuels. And Michael, if those of you who don't know, he's a Nobel laureate. In 1988, he received a Nobel Prize for his work on photosynthesis. So this is a guy who knows a thing or two about photosynthesis, right? And what he said in the talk was basically that the efficiency of photosynthesis is super low. It's less than 1%. And so he said that the sort of area that you need in terms of plants to convert those to biofuels and the sort of solar energy that you need doesn't make any sense. Instead, we should just be focusing on photovoltaics, right? So that was sort of more of a premise of his talk. So I just wanted to take sort of your I want to ask your take on this, that what is your sort of global view on the role of biofuels?
Well, I've known some Nobel laureates who made mistakes as well, but he's not wrong. I mean, the fact is, it is fairly inefficient. Again, as I said, you use water to make them. On the other hand, we have a lot of waste biomass as well. And I think much of the attention has focused on using things, I mean, to make paper, and we have not gone to a paperless society. You have a lot of waste lignin. Lignin is full of aromatic rings, full of energy. Why don't we use that? As it turns out, lignin, a more complex structure that we're working on now, but I didn't talk about, operates, well, has been thought convincingly to decompose by radical chemistry. So bang, we have that. There's some interesting ideas that these things sort of self-catalyzed reactions help convert it. But the point is, we've got biomass, whether you want to depend on it or not. Second, photovoltaics, we don't yet see a path how we can use that as a portable transportation fuel in aircraft or systems like that. There was a wonderful talk at the US National Combustion meeting this in May, where formerly United Technologies, now Raytheon Technologies, is doing a lot of work to how electric can assist aircraft, particularly in the takeoff stage. So it's not foolish to say that we'd rather not. We'd rather have just magic thing, but of course, solar collection is also down about 10%. Also, we can do better jobs there. So there's no question there are disadvantages to biofuels, but I think the driving force tends to be bio oils as far as mass use of it. And one of the incentives is that, again, you can recycle all this waste material in a circular economy of generating, using, then reusing in some fashion.
Yeah, thank you, Phil. I think I agree. I think if we focus on the waste part, then it makes sense. But if someone says that let's grow plants to convert them to biofuels, right? I think that's a difficult proposition to make then.
We can make fun of legislators because they do weird things sometimes, but when I worked for National Science Foundation in 2006 to 9, I got to watch the US Congress debate the issue of whether it is ethical to make biofuels from corn. And they actually debated the ethics of it. I was blown away that they would do this. And they came to the nuanced conclusion that yes, it's unethical in the long term to take foodstuffs and convert them to fuel, but that in the short term, to create an infrastructure for, in particular for lignocellulosic biofuels, that it was ethical to create an ethanol industry using corn, but again, in the short term. So there are these trade-offs, and it's not always necessarily a thumbs down, thumbs up. Sometimes we have to do a balance. And again, with bio oils, my car is a hybrid. It's not pure electric because I have to take trips that are too long for that to cover. I like having that flexibility. I count on it. But I can tell you that with most of my commutes being 30 miles from my home into Raleigh, I go 2 months without putting new gas in. So there's trade-offs. And as combustion scientists, we have special strengths to be able to bring to that discussion.
All right, thank you for that. Sorry, we'll go to Iguang's question. And actually, I had that question in my mind as well. So maybe you can read it in the chat, or I'll just read it for you. That basically,
I can ask it. I probably can stimulate the discussion among the panelists. My question that is very impressive that you showed that in around 2009 or 2010 that you're using this molecular beam mass spectrometers to detect all the species and including radicals like HCO, methyl, and other things. So, and today that there are people in other society for plasma society, they try to using EI, molecular beam, try to detect H radicals, N radicals, and feel very confident to present that to everybody. So my question to you is that as an expert and pioneer in this area, and I want to ask you that what is that uncertainty when you try to using molecular beam, particularly EI and a molecular beam to say I measured quantitatively about HCl, methyl, and OH radicals, and what is that the source of uncertainty from sampling quenching and also that fragmentation, EI process? So just give me some kind of expert thoughts, so share with us. Thank you.
Well, I would say there are at least three different reasons that are causes for uncertainty. One is simply the calibration factor. We can calibrate directly for things like argon, carbon monoxide, hydrogen, and so on. Yeah. We cannot take a bottle of radical and feed it in. That said, it's a powerful technique in combustion molecular beam mass spectrometry to characterize a flame very well, say a hydrogen-oxygen flame, and nail it down not only by calibration factors, but the realization that you've got partial equilibrium in the post-flame gases, and you know the temperature, you can say within a couple of percent what the concentration of radicals are in that, and then use that as a calibration source to do the measurement. How much do you get? So calibration is one factor. The sampling process is what most people worry about, and necessarily, but not necessarily rightly. The reason I'm saying that sort of ambiguously, that so much is determined by the design of the source that you're sampling and the design of the sampling probe. In 1974, 76 or so, Biordi, Lazar, and Papp did this lovely study showing how a typical microprobe will just destroy radicals because there's too many wall collisions. You can't allow wall collisions. What they ended up doing was finding that if you have a wide angle, you don't destroy the radicals, but you do destroy the flame. Yeah. So they came up with a hybrid tip where the design is very critical. The conduct is very, if you have a thin tapering probe, you're asking for trouble because you want the collisions to be with gas molecules, not the wall. It takes 5 to 10 collisions for these molecules to be cooled. Fortunately, most collisions are non-reactive. If you've got non-reactive gas in your skimmer region, which you do because walls quench radicals out that touch the walls, then you can have these non-reactive collisions that cool it down. But the design of the sample probe, Oleg Korbanichov uses a very small little tip on the end of a bulging tip in order to achieve the same sort of thing. So probe design, skimmer positioning is a factor too, but probe design is the second major thing. Fragmentation is something that I think we've worried about. Klaus Hohmann worried about it when he was doing the very original work. And it's very easy to do that. In fact, the first radical measurements that they made of methyl and of C2H, we unfortunately had to show the results of fragmentation at energies lower than the threshold for fragmentation. The reason is from electron ionization, you don't get a specific energy, you get a spread of energies. Plus, there's a spread of energies in the molecules as they come in, not by the time they reach the ionizer, though. There, you've got a energetically homogeneous species going out. There's not too much spread, and that's been confirmed experimentally. But the fact is, with the distribution of energies, you can have a 90% chance of rain, and it still might not rain at all. Or conversely, 10% chance, and it might rain. There are some electrons that are out on the edge. The lowest level molecular beam mass spec signal I've ever gotten was 20 counts of vinyl radical over a 20-minute period, except half of those were acetylenes, even though I was 2.5 electron volts below the threshold for fragmentation, just because it was an acetylene flame, there was a lot of acetylene. So Klaus and I had talked about it, and they very much agreed that you have to be really careful and make sure that you detect any interferences fragments. So it's there's a reason that there are only maybe 10 to 12 flame molecular beam systems in the world, and that number, although the players have changed, that number has been pretty much unchanged since about 1980, when Jim Bittner at MIT with Jack Howard and Marty Zabilski and Dan Siri at United Technologies built a system based on Biordi, Lazar, and Pap's system. So I actually have the shell of that Biordi system in my lab. It was one of the things that got burned up in the moving van when I moved down here, but the shell is still there as a historical instrument. You're welcome to see.
Thank you.
Okay, thank you. Other questions? I didn't see in the chat box, but please feel free to unmute your microphone and ask the question.
So Phil, I have to say that I really enjoyed your lecture. I feel like that I'm hearing that the legacy of combustion chemistry study, as well as the history of the past giants, right? The people who work on pioneers in this area. And it's so interesting. And regarding to that Amir's comment, I'm pretty much with that the Nobel Prize winner. I think that it's much make more sense for now, at least, and put on that solar and wind energy, and how to use that and to improve that make green fuels from water, CO2, anything from there, probably more efficient than grow the crops and making biofuel.
Well, as I told Amir, I have to respectfully disagree while agreeing in measure at the same time. This is not a situation, especially if we are reducing the use of fossil fuels. This is not a situation where we can ignore any sustainable source of energy, and particularly with the attributes that having portable fuels can do. There's so much merit to that. In the movie, the original movie Back to the Future, at the very end, they're feeding banana peels into a portable fusion reactor. Maybe we can go to that. But I think certainly within the next 50 years, maybe longer, we're still going to have the need for portable fuels. And yet, don't put it all in. Remember that the combustion community was the source of a lot of the early solar energy work. Hoyt Hottle was the person who began doing solar energy research at MIT and was a pioneer because he was an expert in radiation heat transfer. So his and Adel Seraphim's book that go through that is instrumental to understanding collection of solar energy into a source. So we in combustion have a whole lot of expertise. That's one thing that I had to constantly make the point at the National Science Foundation, that there tends to be the thing, oh, we've been doing combustion a long time, ever since the first fire was harvested from a lightning strike by a primeval man and woman. So the fact is, though, that our understanding has changed. So if you look at the use of quantum chemistry in manufacturing, in materials, and so much of that came from combustion chemistry interest. Modeling reactive flows came from the combustion community. Using lasers for diagnostics came from the combustion community. Even using large systems of elementary reactions, that sort of modeling and handling, especially stiff equations, came from the combustion community. So we've got a lot of tools at our hand. I do see another question that's just popped up. What changes and modifications are required in gas turbine engines to use biofuels? Not my expertise, and there are other people who can talk more on that, but one approach, and here I think I'm definitely in agreement with the idea that why use biofuels and then convert them to petroleum type products. You can do a perfect drop-in if you convert it to, say, carbon monoxide, hydrogen, or to alkanes, where there are no changes at all used. And tests have shown that. In the United States, the Department of Defense uses a lot of fuels, and they have a requirement that in battlefield areas, you have to use a single fuel for everything: tanks, helicopters, everything. So versatile, they have to have something drop in. So they've done true drop-in tests. But the Navy has been testing, looking at oxygenates. I think the obvious thing is that you have to take into account the different heat of combustion that you get from an oxygenated material that's partly consumed. So that changes that. Otherwise, though, as far as the fluid mechanics of it, if you get to non-ideal high pressures, you can start to get some of the hydrogen bonding happening with some of the oxygenates. But at lower pressures and at higher temperatures, they behave pretty much ideally. But I've already told you more than I know. So I'll have to let others speak more authoritatively to that. Thanks for the question, though.
Thank you, Phil. Any other questions? Okay. If not, maybe we can thank again, Phil. And I mean, especially your comment to the previous question about combustion giving us a skill set is, I think, a very important one, especially for the students in the audience. I think this is a message that we give to our students all the time, that yes, there may be less reliance on combustion going forward, but the skill set that you gain from here is really special and unique and applicable to a variety of fields in future. So I think that's a very important message to come from you as well.
Yeah, I think that chemical engineers preach really strongly the idea that our discipline is not a silo, but it's a core of knowledge that is really transdisciplinary. We learn whatever we need. Mechanical engineers, though, preach the same sort of thing. And I think an ideal for disciplines in this transdisciplinary world is to recognize that you use the expertise that you have in a very far-reaching way. My students don't come in as analytical chemists, but they have to learn a lot about analytical chemistry. A lot of us have had to. They may not come in with very sophisticated fluid mechanics, but they may have to learn to be able to do computational fluid dynamics in a sophisticated way. Well, fine, you have the basic tools, just learn it, or learn enough to be able to communicate effectively with an expert in the field.
Maybe we'll take the last question, which is actually on the same topic we've been talking about already, but it popped up in the chat. You can see it's being asked, biofuel or H2?
Better for future emission, H2, absolutely. And we're learning things that H2 is more complicated than just the simple sort of 19 reaction model that we have too. As we get into different regimes, particularly as we get to high pressure regimes, the business of handling hydrogen. So we also find teams of people, particularly in Europe and starting the US, to look at alternative fuels that are neither hydrocarbon nor hydrogen, like ammonia, the original chemical refrigerant, for example. As a chemical engineer, at first it seems obscene. We spend so much energy and technology to make ammonia from nitrogen that why would you ever want to burn it? And yet there are sustainable ways to do it, increasingly being tested to take nitrogen from the air and hydrogen from water and make it sustainably and use ammonia. There are plenty of problems with adapting to gas turbine engines for using ammonia hydrogen mixes, hydrogen oxygen mixes, and so on. So there's plenty for us to do with good fuels. The ammonia part, I'm still waiting to be convinced, but I'm paying attention. I'm listening to what's being said. Again, it's as Yiguang and I were saying a while ago, it's not a situation where there's one solution solves everything. We have to use many resources at our disposal.
Okay. Thank you very much, Phil, for that answer. Thank you for your time today and for the excellent talk. I really enjoyed it. I'm sure others as well. And as Yiguang explained in the beginning, you'll be sent a link of the talk for you to approve, and then it will be uploaded after that.